first time you heard about the DuPont mercury contamination?
Probably my teen years.
But you've heard people joke about maybe the fact that mercury contamination in fish tissue isn't really a good reason to eat fish from the river.
I've heard that, but I also know lots of people who fish from here that get fish and cook them, so.
In the 1970s, mercury was discovered in a section of the water in the South River located in Waynesboro, Virginia, in response to unsafe waste disposal from the former DuPont facility who was manufacturing textiles at the time. The mercury was initially found in the soil, but eventually eroded into the water of the South River. Ever since its discovery, mercury pollution in the South River has been closely monitored. To investigate these topics, we headed to Waynesboro to measure snail populations. Since mercury is toxic to most organisms, we hypothesized that snail populations upstream of contamination would be larger than those downstream.
To estimate population sizes, we used the mark recapture method. We established 4 capture sites upstream and 4 sites downstream from DuPont, each site being 1 square meter in area. At each site, we counted all the snails we captured and marked them with fingernail polish. Then a week later, we came back to those same sites and counted again. This time, we noted how many snails were marked from the previous time and how many new unmarked snails were present. These data were then plugged into the mark recapture equation, which gave us estimated population sizes.
The results of our experiment were ultimately inconclusive. The average estimated population sizes, as derived from the mark recapture equation, were 1,609 snails downstream and 1,201 upstream. Because of negligible difference between the two population sizes and high degree of variability, as displayed by the error bars on Figure 3, the population sizes of upstream and downstream snails could not be determined to be different.
Thus, we could not accept our initial hypothesis. A major limitation of this study, which may have restricted our findings, was small sample size. Because we only collected from 8 total sites, our population size estimates were likely subject to a high degree of random error.
The results of this research are not senseless, however, as we have highlighted the dangers of mercury pollution in the South River. Further, we have addressed the public concern with local polluted water and reviewed methods for remediation. The ongoing struggle to combat mercury pollution is gaining attention, and so consequently, public officials have already begun ranking remediation alternatives that balance efficiency and cost effectiveness.